My name is Dan Liebl. I am a professor of neuroscience at the Miami Project to Cure Paralysis and the co-director of the MD PhD program at the Miller School of Medicine. So the most exciting science in my lab currently is a project I developed about 8 years ago with colleagues around the world. And the project involved what is really the underlying question, or what is really the problem that leads to dysfunction in humans. For 40 years, we've tried to focus on the injury site itself. And we looked at different injuries like spinal cord injury, traumatic brain injury, and there's been a lot of trials that have failed. And we decided to step back from that and say, what is really happening within the brain? And one of the things that we found out was that along with the damaged tissue that we see, there's a lot of dysfunction that goes throughout the brain. And most of that is the result of synapses being deinnervated, so synaptic losses. So recently we've discovered that one of the reasons, or asked the question of what is the mechanism that leads to this synaptic dysfunction? And we found that this unique amino acid called D-serine, which is one of the few D amino acids, since most of our body contains L amino acids. It plays an active role in how the brain works. In pathological conditions like traumatic brain injury, this amino acid is expressed at high levels for long periods of time. And this overexpression leads to synaptic damage. And we're in the process of developing therapeutic strategies to block this effect and to improve outcome of the synaptic damage throughout the brain. We think that the therapies that we're developing with pharmaceuticals can be applied to patients while they're enter the hospital, so in the OR. We've discovered that if they're given during a short segment of period for the first several weeks, it can have long-term recoveries. And we think that this will be a very viable solution to block the dysfunction we see in humans. When I was a postdoc many years ago, I worked with a group called the Christopher Reeves Paralysis Association. I was one of the first members of this consortium that brought people into the injured field, and from that day forth, I became infatuated with trying to find a cure. And I was recruited to the Miami Project in 2000, and my whole career has existed in the framework here. And I believe that it's been the best choice of my life.